This is my project about cloning animals by Karina Garcia. The definition of cloning an animal is the technique of making genetically identical copies of the original organism. The scientists use the DNA of the original organism and implant it into a surrogate who then produces an almost identical organism. Cloning animals is a biotechnological process involving the replication of an existing animal's genetic material to produce one or more genetically identical individuals. So, how does cloning work? Cloning an animal is a long process of producing an organism with the same genes and DNA as the original organism. Scientists can use any DNA that they can find from the original organism besides sperm or egg cells to clone the animal. Scientists gather DNA from the organism's somatic cell and transfer it into the surrogate's egg cell. The nucleus and DNA are removed from the egg cell through this process. The somatic cell must be from an adult cell and can be from anywhere including the skin. Before the scientists can clone the animal, they must remove the nucleus from the surrogate cell. The biology of cloning animals involves the process of the somatic cell nuclear transfer, otherwise known as SCNT. There is multiple steps for SCNT. First step of SCNT is, of course, donor cell collection. This is where a somatic cell is taken from an animal to be cloned. Somatic cells are any cell in the body other than reproductive cells. The second step of SCNT is egg cell acquisition. This is where egg cells are collected from a female animal of the same species, and these eggs are usually from the ovaries through surgical extraction. The third process is nuclear removal. This is the process of removing a nucleus and leaving behind an enucleated egg cell. This is crucial because it clears the way for the insertion of the donor's cell's nucleus. The fourth step is donor cell fusion. This is where the nucleus of the donor somatic cell is extracted and inserted into the enucleated egg cell. They can do this by using a very fine needle to carefully transfer the nucleus into the egg cell. The fifth step is cell activation. Cell activation occurs if and only if the nucleus is successfully transferred into the egg cell, where the two cells are fused together. Scientists either use chemical or electrical stimulation that is applied to trigger the egg cell to start dividing, mimicking the process of fertilization. The sixth step is embryo culture. The fused egg cell, now containing the genetic material of the donor somatic cell, starts to divide and develop into an embryo. The embryo is then developed under a laboratory under controlled conditions to support its growth. The seventh and final step is embryo transfer. After several days of close observation and development, the embryo is then transferred into the uterus of the surrogate of the same species. The surrogate then carries the embryo to term that gives birth to a clone that shares the same genetic material as the donor animal. For this to work, it requires manipulating cells and having careful control of the developing stages. This process involves cell culture, egg manipulation, and embryo transfer. It must be good quality donor cells, healthy and viable eggs, and the donor nucleus and surrogate must be very compatible. The cloning process is very complex and success rates vary depending on the species. For cloning, mammals have been the primary focus of animal cloning research, and species such as cattle, sheep, pigs, and dogs have successfully cloned using SCNT. Generally, cattle and sheep have had higher success rates to pigs and dogs. Cattle and sheep cloning have achieved success rates ranging from 1% to 5%, while pigs and dogs may have lower success rates. Rodents have a higher success rate than mammals, with having 2% to 5% chance of being successful. In non-mammalian species, their success rates have been much lower than mammals. They face challenges associated with their reproductive biology and embryo development, which impacts their cloning efficiency. Primates have had a much harder time being cloned due to their reproductive biology and ethical considerations. There have been some successful attempts at primate cloning, but the success rates still remain relatively low. A cloned animal's death age has nothing to do or correlate with the original animal. Factors such as environmental conditions and the health of the animal. The animal's diet and health care also impacts the animal's lifespan. It may live longer than the original one, but it just depends on its lifestyle and if it has any diseases. The cloned animal and the original animal both share the same genetic blueprint, though they are not the same exact replicas. Factors such as diet, living conditions, exposure to toxins, and stress levels can profoundly impact in lifespan. While they do share the same DNA as the original donor, they lack genetic diversity when they try to produce their own offspring. The lack of genetic diversity can limit the clone's ability to adapt to environmental challenges and increase its susceptibility to disease and other stressors, which thereby affects their lifespan. There is a theory that the age of the donor does indirectly influence the lifespan of the clone through mechanisms such as telomere length and cellular senescence. Telomeres, which are protective caps at the end of chromosomes, shorten with each cell division and are associated with cellular aging. Older donors may have shorter telomeres, which can indirectly impact the clone's lifespan. Dolly the sheep was the first ever animal cloned. She was created from an adult cell, and she only lived to about 6 years old until she died from a lung infection. She was born on July 5, 1996, at the Roslin Institute in Scotland. It took scientists a result of 277 attempts at cloning using the somatic cell nuclear transfer technique. Out of all of these attempts, only one embryo successfully developed to term and was born as Dolly. Bioethics of cloning animals causes harm to all the organisms involved. Companies who promote or clone animals exploit the grieving owners. Some people might say that the scientists who clone animals are playing God. It can take hundreds of attempts to finally produce an offspring, for example, Dolly the sheep, which took 277 attempts. The cloning process may involve physical harm and suffering for both the donor animal and the surrogate mother, as well as potential health issues for the cloned offspring. Cloning animals can potentially compromise species' resilience to environmental challenges such as disease outbreaks or climate change. The loss of genetic diversity can cause detrimental effects on the ecosystem stability and biodiversity. Although it may improve livestock, it still declines the genetic diversity within the animals. Cloning animals for biomedical research purposes raises ethical questions regarding the use of animals in scientific experimentation. If scientists do use these cloned animals for their experimentations, the animal may experience suffering with physical and mental issues. People might use this process to clone their pets. Although they might think this is a good idea, their pet might not come out the same as it was before. It could have a completely different personality or look completely different. Because cloning animals is so new, there might be birth defects or neurological disorders that the original pet did not have. Cloning animals contributes to the objectification of animals as mere commodities. Pet cloning may divert resources and attention away from other pressing issues in animal welfare and conservation. The decision to clone your pet must come down to a careful consideration of ethical principles, scientific integrity, and the best interest of both animals and humans involved. Companies can exploit the idea of cloning your animal as a way to get money from grieving owners. Advertising pet cloning can tap into the deep emotional attachment that many people have with their pets. Pet cloning can delve into the deep emotional attachment that many people have with their pets. Many companies offer the possibility of bringing back a loved pet through cloning. They can appeal to the desire for continuity, closure, and consolation in the face of loss. Grieving owners face emotional vulnerability, making it easier for marketing strategies to get through to them and having them clone their animals. Companies might capitalize on the exclusivity and novelty of pet cloning because it can be marketed as premium, high-end service and catering to individuals willing to pay a premium for the opportunity to clone their beloved pet. This marketing approach exploits the intersection of emotional attachment and consumerism, enticing grieving owners with the promise of a unique and personalized experience. Cloning animals is a bad idea, as there are many animals in shelters or on the streets that deserve a home. Cloning mentally and physically hurts the surrogate and the donor. It can increase the amount of animals on the streets. It can take hundreds of times to finally produce the organism. For example, Dolly the sheep, which took 277 times and only produced one organism or embryo that survived. It wastes resources that can be used for other researches. Although some may argue that it provides an opportunity to preserve a beloved pet, others raise concerns about the welfare of the cloned animal and ethical considerations surrounding manipulation of life. Ultimately, it depends on the individual perspectives and values. Before a person goes and clones their animal, they should research about how cloning can mentally and physically hurt the surrogate and the donor. On the side that is for cloning your animals, they believe that it has the potential to alleviate the pain of losing a loved pet. For many pet owners, the bond with their animal is very deep and they feel like it is one of their own family members. The ability to clone a pet may offer a sense of relief, allowing the individuals to experience the bond anew with a genetically identical animal. On the other side, cloning can involve invasive procedures for both the donor animal and the surrogate mother. This raises concern for the physical and physiological well-being of these animals. The cloned animals may also experience health issues due to genetic abnormalities or complications during the cloning process. As animal cloning becomes more and more popular, it diverts the attention and resources away from animal welfare, such as overpopulation, homelessness, and improves the lives of existing animals in shelters. If we start investing in cloning technology, it may perpetrate the idea that animals are replaceable commodities rather than emphasizing the importance of responsible pet ownership, adoption, and supporting initiatives to help animals in need. The black-footed ferrets were going extinct, but a group of scientists decided to clone a few ferrets. There were 7 original ferrets that were cloned. Due to the ferrets being cloned, they face almost no genetic diversity. One of the original 7 was taken from frozen samples all the way back in 1988. They are hoping that 2 of their grown ferrets produce offspring to increase the genetic diversity, and are also hoping that they can develop a resistance to diseases that they had none before. Since the black-footed ferrets face almost no genetic diversity, they often have many health risks. This is due to them being almost identical to each other and having to breed with each other. If scientists were to introduce genetically compromised individuals, this could harm the conservation efforts rather than help them. They might have chromosomal abnormalities or mutations which can lead to health problems in cloned animals. These abnormalities may affect various aspects of the ferret's biology, including development, growth, and organ function. Cloning can also lead to alterations in gene expression patterns that can influence an animal's phenotype and health. These changes can affect the ferret's susceptibility to diseases, metabolism, and other physiological processes. They can also face reproductive issues such as infertility or reduced fertility. Cloned animals may have a compromised immune system, making them more susceptible to infections and diseases. Is there a chance that we could clone dinosaurs? Realistically speaking, it's almost impossible to clone dinosaurs, as extracting intact DNA from a dinosaur millions of years old would be nearly impossible. DNA gradually breaks down over time. Cloning an extinct animal requires a close relative to carry the embryo to term. There is also no guarantee the animal will look anything like the original species. For now, trying to clone a dinosaur will just be a fantasy until we can finally find a way to clone the DNA from their fossils. One of the primary challenges that we face is the preservation of DNA. DNA is very fragile and is a molecule that degrades over time, even under the most favorable conditions. The oldest DNA samples that were recovered to date are around 700,000 years old. This is way younger than the age of dinosaurs, which became extinct about 65 million years ago. Also, the chances of finding intact dinosaur DNA are very, very slim. Even if scientists were able to recover intact dinosaur DNA, it would be very hard to reconstruct a complete genome. DNA from fossilized remains would likely be highly fragmented and contaminated, making it exceptionally difficult to piece together an entire genetic sequence accurately. Even if we were somehow able to complete a genome, finding a suitable host organism for gestation presents yet another obstacle. Dinosaurs and their closest relatives have been extinct for millions of years, and there are no living organisms capable of serving as surrogate mothers for dinosaur embryos. Even though advances in sequencing technologies have made it possible to extract and analyze ancient DNA, the task of reconstructing a dinosaur genome from such fragmented and degraded samples remains daunting. The biological and physiological differences between dinosaurs and modern animals further complicate the feasibility of gestating dinosaur embryos in surrogate hosts. Here are some fun facts about cloning animals. Cloning animals has been used to maintain the genetics of prized animals. Some livestock have been cloned for their desirable traits, such as high milk production in dairy cows, superior meat quality in beef cattle, and specific genetic lines in sheep and pigs. Horses have also been cloned to replicate champion racehorses, show jumpers, and other high-performing horses to preserve their genetic lineage and athletic abilities. Endangered species have also been included in the genetic cloning. Cloning endangered species still remains a subject of debate because of the limited diversity. Examples of endangered species include the gaur, wildcat, and other potentially dwindling populations. While not as common as in livestock, dogs have been cloned and used in the dog breeding world to replicate exceptional individuals such as service dogs, working dogs, or beloved pets with desirable traits. Some scientists clone endangered species, even though it remains a subject of debate because of its limited genetic diversity. Pet cloning has also been introduced, although it is still in the testing progresses and only for people with high-paying jobs. Cloning animals may allow desired traits to be passed down to multiple generations. Although it isn't technically morally right, it can be used to study genetic diseases and develop new treatments. Scientists have also been able to clone body parts and organs for people. Scientists have explored the possibility of using cloning techniques to generate organs for transplantation to address the shortage of donor organs. They have been able to make skin grafts for treating burn victims and patients with severe skin injuries. By taking small samples of skin cells from a patient and cloning them to produce additional skin tissue. They have also been able to make cartilage, which plays a crucial role in supporting and cushioning joints. By replicating cartilage cells from a patient's own tissue, researchers aim to create personalized treatments that minimize the risks of rejection. They have also been able to produce nerve cells, including neurons and glial cells, for studying neurological disorders. Here's a quick review of what you have learned through this video. Cloning animals causes harm to both the donor and the surrogate. It uses the CNT method, which is somatic cell nuclear transfer. You cannot use reproductive cells in SCNT. It must be an adult somatic cell. Dolly was the very first cloned animal back in 1996. It may take hundreds of attempts to accomplish getting an alive embryo. We cannot clone dinosaurs just yet.